Hello guys, welcome to another video. Today I'll be teaching you all Lac Operon. So Lac Operon, it is a concept. It is in the genetics chapter, the second chapter, molecular basis and inheritance. So generally this is not really a difficult topic, but it can be a confusing topic if you don't do it properly. So today we'll be looking through it. I will explain it to you all, and I will also be writing the notes side by side. Last video I made about ETS in respiration. You can check that out too. So in that I didn't really write much notes. I had it in my book, but I didn't write it down on the board. Today I will be writing it down. So let's just start out with the basic introduction. Then we will dig into the main or the deep stuff. So let's start. So first it was given by, this is very important. You can learn the whole Lac Operon concept, but if you don't know this, then you might not be able to solve a question which might come in the next year's exam. And NEET 2025, it is gonna be a bit of difficult, on the tougher side you can say, so you might as well do it. So this is given by genetist Francis Jacob and biochemist Jacob Monod. Now, as important the names are, so is the qualification. Geneticist, and this was biochemist. Who was biochemist? Jacob Monod, and Francis Jacob was genetist. Now, they were the first to figure out the transcriptionally regulated system. So what does that mean? So transcriptionally regulated system is in which the process of transcription, which we have read in your before pages, that is a regulated system. So how is it regulated? We will learn that in this Lac Operon, but they were the first to discover that. So the question can be formed in two ways. It can either come who invented, or like who not I'm sorry who discovered Lac Operon, or who were the first one to figure out transcriptionally regulated system. So these are the two ways by which the question can be framed. So yeah. Now, what is Lac Operon? So it is a polycistronic structural gene. We will dig into it deeply, but just for now, just remember it is a polycistronic structural gene. It is regulated by common promoter and regulator genes. It is regulated by common promoter and regulated genes. The lac in Lac Operon stands for lactose. Very important. It stands for lactose. Just give me a second, I will write. So guys, as we talked about, let's start with Lac Operon. So Lac Operon basically has one regulatory gene and three structural gene. Now just remember how I'm telling you to remember it. Just remember Lac Operon, it is divided into two parts. It has one regulatory gene and three structural genes. Now one regulatory gene, what is the regulatory gene? It is the I gene. Remember, I gene. What is I gene? It is the repressor of Lac Operon. Now, what does the repressor of Lac Operon means? Basically, Lac Operon is trying to do transcription. So, what is it? What is it trying to do? It is trying to do transcription, and this I gene, it stops it from doing transcription. So basically, it is repressing it. Ki you cannot do transcription. It is stopping it. So that's why it is called repressor, or we also call it as inhibitor. It inhibits it inhibits the Lac Operon to not do any transcription. So another important line, it is given in NCERT. It is an inhibitor, not an inducer. As we go further, we will learn that there is also an inducer, which is lactose. We will learn about it later. But important difference, I here stands for inhibitor, not inducer. It can come in a statement based question that in Lac Operon, I gene stands for, they can give that I gene stands for inducer. So that statement will be wrong. What does I gene stands for? It stands for inhibitor. It stops it from happening. So this is the regulatory gene, and there are three structural genes. What are the three structural genes? There is Z, Y, and A. This is very simple. You will learn this, like if you revise it like 4 or 5 times after I teach you, you make your notes, you can revise it within like 4 or 5 times. This will be very clear. So three structural genes, there are Z, Y, and A. Z transcribes beta galactose. There is a full name, beta galactose. It is called B-gal in short form. Y gene transcribes permease, and A gene transcribes transcriptylase acetylase. So what does BGAL do? It initiates the hydrolysis of disaccharides. Now, if you have read biomolecules chapter in your 11th standard bio, or you have read it in organic in the chemistry in 12th standard, you know hydrolysis of disaccharides turning lactose. I don't know if you can see, turning lactose into galactose and glucose. Means lactose is disaturated form, and galactose and glucose, these are monosaccharides. So it breaks lactose into galactose and glucose. Very again very important. It can come that BGAL, what does it do? Hydrolysis of what? So it hydrolysis of lactose, and it turns lactose into what? It can turns lactose into galactose and glucose. Initially this is a very easy question, but since the paper is coming like that, it can come. So please remember it. It turns into galactose and glucose. So now permeases. What does permease do? It increases the permeability of BGAL for the cell. मीन्स ये जो BGAL है, ये क्या करता है? हाइड्रोलिसिस ऑफ डिसैचुरेट करता है, और परमियस क्या करता है? उसकी कैपेबिलिटी और बढ़ाता है. मतलब ये हाइड्रोलिसिस ऑफ डिसैचुरेट को और प्रमोट करता है. सो जेड जीन, व्हाट डज इट डन? BGAL hydrolysis of disaccharide it does, and why it promotes it, it increases the permeability. मतलब this Y promotes the Z. Okay, the function of Z is something, and Y, what is it doing? It is promoting. Okay, so that, and A, just remember, A transcriptase. Not much information is given about the transcriptase, so you can just remember that. Now genes, another important statement given in the NCERT. Genes are needed to function, look here, genes are needed to function in same or related metabolic pathway. Means these three structural genes ZYA, they cannot perform in a separate metabolic pathway. They have to perform in the same manner or in a sequence manner. If you have read Krebs cycle in botany, or many other Kelvin cycle, all the steps, they happen in sequence. Now that is what the statement is trying to say. It is telling that everything has to be in sequence, everything has to be in the same metabolic or related metabolic pathway. It cannot be different. It can again come in a statement based. Now all three structural genes are required for metabolism of lactose. So what is the main purpose of the Lac Operon? It is metabolism of lactose. Now I have written in the back slide that Lac stands for lactose. So main Lac Operon purpose is metabolism of lactose. Now all three genes are included in the metabolism of lactose. A statement can come that only Z, it can say that the Z gene is only involved. The Z gene is involved in Z gene is the only gene which is involved in the metabolism of lactose. Now it is completely wrong. As you can see, the main metabolism of lactose is done by Z gene, but Y gene and A gene also help it. We saw here, right? It increases the permeability. So all three are required for the metabolism of lactose. Okay, so now we will get into another slide. I hope it's clearly visible. So now lactose. Lactose is a substrate for enzyme BGAL. Now if you have read biomolecules, another very important chapter, in the last there is an enzyme substrate and all that. It forms enzyme substrate, then enzyme product, and then it releases, all that stuff is there. So the main thing you have to remember, lactose is a substrate for enzyme beta galactose. Another important question it can be asked in the exam. Lactose is a substrate, and it is for the enzyme beta galactosidase. Now what is lactose? It is an inducer. Again very important. What did we learn in the previous sign? I gene was inhibitor. What did we learn? That I gene was inhibitor, lactose is inducer. Means for that Lac Operon process to has, or for the metabolism of lactose, what do we need? We need lactose. So lactose in the inducer. Because of lactose the processes happen. Now it regulates on and off of the operon. Another very whatever I have written, you may not understand it completely. It will take some time as you learn and as you analyze the process, but these are very important facts you have to learn. Who regulates on and off the operon? Lactose regulates on and off the operon. Simple seedha question can come in exam. You don't have to think much, just learn this. So who regulates on and off of the operon? It can come in options, I gene or any enzyme they'll give, but you have to tick lactose. Lactose regulates on and off the operon. Now another very important question. Now this came in neet, the year I gave neet, this came in neet. If carbon source is absent, which is the common carbon source? It is glucose. If carbon source is absent, then lactose is transferred into the cell through the action of permease. Now see, if carbon source is present, then it allows the lactose to go inside the cell. Now see, you don't have to go do PhD in this to completely understand this. I had to like read it like 10 12 times to like completely understand it. But the thing is, the chances, only one question has to come, and for that, if you even learn this, it's fine. So if carbon source is absent, remember, when does permeases show action? When carbon source is absent, then lactose is transferred into the cell through the action of permease. When I gave NEET, this enzyme was asked. The question was, if carbon source is absent, then lactose is transferred into the cell through what enzyme? So it was permease. Now, always remember, a low level of Lac Operon has to be there in order for lactose to enter the cell. Means that the metabolism of lactose is occurring. The Lac Operon is performing its function, but in the cell, actually, a low amount of Lac Operon should always be present. Like since the process started, it always has to be, a low amount is always has to be present. Now again, it can come in an assertion reason that a low level of Lac Operon has to be there in order for lactose to enter the cell. Without it, the process will not continue. So it will be true. Small amount is always required. See, it will. They can also give you a statement that no level of Lac Operon has to be there for this. So it is completely wrong. Low level has to be there. Okay. So that is that. Thank you. Okay guys, so now we will start the main mechanism of Lac Operon. So I will, before reading all this stuff, I will give you a brief idea of what exactly Lac Operon does. So the main job of Lac Operon is to do the transcription or the metabolism of lactose. Now what is, as we read in the before slides, I have showed you that I gene is the inhibitor. There are three structural genes, then lactose is the inducer. So what does, so main we will here focus on lactose and I gene. I gene is what? I gene is the inhibitor, so it will inhibit the process, it will inhibit transcription. Lactose is what? It is inducer, so it will induce the process of metabolism, or it will induce the process of transcription. So they both come in cross. So what happens when lactose comes in contact with I gene? So it metabolically deactivates I gene. Okay, so what happens when in to state you in simple terms, when lactose and I gene come in contact, lactose kills the I gene. So because of that, transcription occurs. So that is basically, that's it. That's basically the process. So I gene produces a repressor constantly. So what is I gene is producing the repressor constantly. The repressor protein binds to operator region of operon and prevent RNA polymerase from transcribing the operon. So what happens when lactose is not there? Isen is producing the repressor constantly, and the repressor goes, and there is like a operator region, like a strand. There is operator region in that. So what happens is the repressor goes and connects with operator region. So because of that, it prevents RNA polymerase from transcribing. Now you know RNA. You have done transcription before this. So transcription में क्या होता है? RNA polymerase is responsible, enzyme responsible for transcription. So what happens, because this repressor binds to the operator region, it prevents RNA polymerase from transcribing the operon. ओके, सो जैसे ऑपरेटर रीजन है, वहाँ पे आरएनए पॉलीमरेज को जाना था, पर अभी क्या हो गया है, वहाँ पे जाके रिप्रेसर बैठ गया, तो आरएनए पॉलीमरेज कैसे जाएगा? तो नहीं जा पाएगा. दैट्स वाई इट अवॉइड्स ट्रांसक्रिप्शन. So this is basically very easy if you have read the again I'm saying it, but if you have in biomolecule chapter, if you have read the enzyme or the what enzyme substrate part, enzyme complex and all that, it will be very simple for you. You will be able to understand. But even if you don't, you will get the idea of what I'm trying to say. So when an inducer, lactose or allolactose, again in NCRT in the starting line it is given that in Lac Operon inducer is lactose, But when this statement was given, I've picked up the statement straightly from NCRT. In bracket, it was given lactose as allolactose. So in exam, it was come that which of the following is an inducer. So first option they can give lactose, second option they can give hygiene, third option they can give allolactose, fourth option they can give A and 3 both. Now, as a student, you are in a hurry to complete the paper because you have to do physics, you have to do chem as well. But please read all the options. So see, inducer, you have in your mind that yeah, lactose is inducer. You will take the first option and move on. But in bracket it is given the lactose or allolactose. So lactose and allolactose both are inducer. So you have to take the fourth option, which is 1 and 4 both. So you it is general tendency. You will see first option it is lactose, you'll take that. Then you will check second option. Okay, hygiene. Okay, it hygiene to nahi hai. It is inhibitor, and you'll please check all options. It is lactose as well as allolactose. So when inducer lactose or allolactose interacts with the repressor is inactivated, just as I told you. So when inducer lactose or allolactose interacts with the repressor, it inactivates it. So because of this, what will happen? Because of this, transcription will take place. RNA polymerase can take place. So now this allows RNA polymerase access to the promoter and transcription occurs. So just as I said, so the repressor is removed from the operator region. So this allows RNA polymerase access to promoter and transcription occurs. Regulation of Lac Operon is same as regulation of enzyme synthesis by its substrate. This is in biomolecule. Very important chapter. So regulation of Lac Operon is same. Okay, this was I've been telling you. If you have read this, then it will be easy for you to understand. This is given in NCRT. It won't come in exam as a statement, but it is given in NCRT. And another important, this will, I guarantee you, this will come whenever you're watching. I don't know if it has come but I have very gut feeling that this will come. Regulation of Lac Operon by repressor is called as negative regulation. Now there is positive regulation and negative regulation. I can explain positive regulation to you, but it is really not relevant at an 11th, 12th basis. It was given in my coaching modules. I read it, but NCRT में it is not given. So please don't do it. NCRT is बस NCRT का तो बहुत हो गया. Regulation of Lac Operon by repressor is negative regulation. Remember. और ये भी हो सकता है, you will like remember that it is negative regulation, but regulation of Lac Operon by repressor, they can give you inducer, they can give you anything, repressor or maybe inhibitor ऐसा बोल दिया तो it is negative regulation. So that is the very important diagram. So that is the very important point. Now I will draw the diagram of what it is given in NCRT, and I will explain you the same process again. If you want, you can skip that part, but NCRT में diagram दिया है, तो I'll draw a diagram and explain it to you as well. Okay guys, sorry my audio went bad a little seconds ago. So don't worry about it. I will explain the everything process again to you. So now as in the diagram we can see that I gene, it produces the repressor mRNA. Now this is the repressor produce kya. Now what happens, the repressor goes and binds to the operator region. This is the operator region. So ये क्या करता है, जाके operator region पे करता है. Then RNA polymerase, say this is a stand for RNA polymerase. So this cannot come here. नहीं आ पाता ना, क्योंकि ऑपरेटर पे रिप्रेसर बैठा हुआ है. सो व्हाट हैपन्स व्हेन इंड्यूसर इज प्रेजेंट. दिस इज द इंड्यूसर. व्हाट इज दिस इज लैक्टोस acts as inducer. It goes and binds with the repressor के साथ bind करता है, and then what does it form? It forms an inactive repressor. So this now cannot go at the operator because ये आ गया है. inducer has come and it has deactivated it, as I explained in the previous slide. If you increase the volume to top and you have earphones, you can still hear me. Actually my earphones disconnected, sorry about that. So now what happens when lactose this happens when lactose is not present. When lactose is not present, repressor goes and binds to the operator region. When lactose is present, when inducer is present, it binds to the repressor and it forms inactive repressor. So now these are free to produce lac mRNA or lactose mRNA, or continue the metabolism of lactose or the transcription of lactose. If you have any problem audio of the before, if you want to listen it very clearly, then please write in the comment section. I will again make a video on this, on that specific part. But I think I gave you all a basic idea of what this is. I wrote every point in my slides, so even if you make notes from I strictly say read NCRT, read it 10 times, this lac operant word, read it 10 times. But even if you make the notes from whatever slides I have given, I think it will be more than enough. Now the main thing is to understand concept, because they will definitely give you statement based question as well, so please understand the concept as well. So thank you, please like and please subscribe, and please let me know what other topics you want me to make videos. I understand that a whole chapter is never difficult. There are some topics which are difficult, so you can write it down in the comment section and I will definitely 100% make video on them. Please subscribe, these are very early videos, so I will really appreciate the support here. And if you want me to improve something, or make videos more in Hindi or completely in English, or whatever your suggestion is, please write it down in the comment section. Thank you for watching the video. Please like, take care, bye, and good night. It's the night now. Good night. Thank you.